Hello and welcome to the Apologetics Academy. If you're joining us for the first time, this is a weekly webinar which I run normally on Saturdays. This week we're running on a Monday, and we run at 8 p.m. UK time, 3 p.m. Eastern time, 2 p.m. Central time, and 12 noon Pacific time. And each week we bring on different scholars, thinkers, apologists, scientists from across the theological and philosophical spectrum to present on topics that are of interest to Christians, particularly on topics that pertain to the question of whether or not Christianity is true. And we also cross-examine and evaluate competing worldviews, including Islam, Mormonism, atheism, and so on. If you are joining our webinar on Zoom this evening or today, where you are in the world, there are a number of ways you can participate. You can simply watch and listen anonymously if you so choose. There's also a chat window where you can type in chat. Please, if you're doing so, direct chat to everyone rather than just panelists. You can also submit questions in textual form anonymously if you wish by hitting the Q&A button at the top of your screen. And I will, at the appropriate time, read that question to our guest speaker, and he will do his best to respond. You can also click the raise hand button, and I will promote you to be a co-panelist with our guest speaker. Please bear in mind that this session is being recorded. Just bear that in mind if you are participating. Also, you can, just to give you an idea of what's coming up in future weeks, this coming Saturday, May 20th, we have Jonathan Docherty of Broken Ministries, who is going to give us a presentation on God's design for sexuality, and give his own testimony of God's grace in his own life, from bondage to sexual addiction to freedom and joy in Christ. Then the following week, on May 27th, we have Dr. David Wood of AnsweringMuslims.com and Acts 17 Apologetics, who's going to be talking to us about the psychology of Islam. And then the following week, on Saturday, June 3rd, we have Dr. Jeff Zwierink, who is an astrophysicist who works with Reasons to Believe, who is going to be giving us a presentation on the topic, is the multiverse an information generating machine? So we have some very interesting topics coming up over the next several weeks. Just returning to today's session, then, we have with us, we're very honored to have with us Dr. Ola Hössjer, who is a population geneticist and mathematician at Stockholm University, and he is going to be presenting for us his recent work on developing a new population genetics model for human origins, and he's going to be asking whether it's possible or plausible that modern humans are descended from just two individuals. So, Dr. Hössjer, do you want to just maybe, for the benefit of those who haven't come across you before, just introduce yourself, tell us a bit about who you are, and then launch into your presentation? That would be awesome.
So, thanks a lot, Jonathan, for the invitation to give a talk here at the Apologetics Academy, and it's a great pleasure for me. And as Jonathan said, I would like to start saying some words about myself. I'm Swedish. I live outside Stockholm. I'm 52 years old. I have two daughters, aged 19 and 21. And my field of study is mathematical statistics, and that's a subject which has sort of two legs. The mathematical part is using probability theory to study models of random phenomena and chance, and using that for statistics and draw conclusions from data. And I became professor of mathematical statistics when we lived in southern Sweden. I worked at Lund University at that time, and that was 17 years ago. And about that time, I also got interested in using statistical methods in genetics, and in particular epidemiology, inheritable diseases, and look at risk genes for various forms of inheritable cancer and diabetes. And actually, the interconnection between statistics and genetics has a long history. It started already in the 1860s with the work of Mendel, when he proposed the concept of genes and his laws of inheritance from parents to children, they were actually framed in terms of probabilities. In the 1880s, Francis Galton, he compared traits, for instance, body length between parents and offspring. And then he invented the concept of correlation coefficient, which is very much used today. Mendel's theories, they were forgotten for a while, but they were rediscovered in the early 20th century. And then genetics developed quickly, and this spilled over to statistics. And many statistical concepts were actually born out from genetic applications. And in particular, 100 years ago, Ola Hössjer, he came up with many statistical, new statistical ideas. And many people, including me, think that he was the best statistician of the 20th century. Yet he was professor of genetics, and most of his statistical work, as I said, was born out of genetic applications. And he was, for instance, the maximum likelihood method when we estimate parameters from a model. So I sort of got into this while I was at Lund University, doing genetic work in epidemiology. Then 2 years later, 2002, I moved to Stockholm and started working at Stockholm University. And then my interest in genetics continued, but I started working on population genetics. And what is that? Well, whereas Mendel's laws of inheritance are sort of within families, they tell how parents transmit their genes to their children. In population genetics, it is the genetic makeup or the genetic composition of a whole population that one studies, how it changes from one generation to the next, and so on. And after coming to Stockholm, I sort of started, after a while, cooperation with conservation biologists, and they want to prevent species from getting extinct. And an important part of that is to keep them viable. For instance, prevent inbreeding in species, because that could give rise to inheritable diseases. And here, population genetics comes in as an important tool in order to study how inbreeding could be accumulated over time. So I sort of started to study population genetics theoretically and in cooperation with my colleagues from the biology departments. And population genetics also has a lot to say about Darwinian evolution, because although Charles Darwin, he published his Origin of Species in 1859, I would say that it was not until in the beginning of the 1940s it became a widely accepted theory, and that was due to work of population geneticists in the 20s and 30s, or quite a bit due to that. In particular, three very prominent British scholars, Ola Hössjer, I already mentioned, Sewell Wright, and J.B.S. Haldane. And what they did, they synthesized Darwin's theory of natural selection and the survival of the fittest. He gave sort of, they gave genetic mechanisms how natural selection could operate. And they tried to explain it in terms of genetics. And I think population genetics is an excellent tool for describing small genetic changes. So the main question is, how much can it explain? And I will come back to that question later in my presentation. So before doing that, I would like to say some words of how I got into the evolution design debate. Because when I grew up in Sweden in the 1970s, I was sort of taught in school that evolutionary theory was more or less an established fact. But I'm also Christian, and I sort of felt a bit uncomfortable to interpret scripture in light of evolution. But since I was sort of, I thought that evolutionary theory was more or less proved, I sort of tried to interpret the first 3 chapters of Genesis more metaphorically, the first 2 humans, Adam and Eve, and the fall of man, and so on. But when I came to Stockholm and started this work in population genetics, I also came across some literature in intelligence design and some creationist literature. And then when I started to read that, I found that there were in fact big gaps of evolutionary theory, because it had difficulties to explain macroevolution. How is it possible for small stepwise genetic changes to explain formation of completely new species with birds with wings, species with new kinds of eyes, photosynthesis, and so on. So, and when Fisher, Wright, and Haldane, when they were working in the 20s and 30s, much less was known about the complexity of cells, for instance. And I still think that the theory that they initiated, and it has developed since then, population genetics, it's still an excellent tool to describe microevolution. And that is changes within species, and that's what conservation biologists are using it for, and some limited species formation. For instance, that all cat animals could have a common ancestor, and all dogs and wolves and so on. But I think nowadays, and that's my view, I think population genetics can help us to understand the limits of evolution. That is, that it seems difficult to go beyond microevolution to macroevolution. So that was sort of the introduction, my own background, and this brings me to the topic of this talk. I will describe, as Jonathan said, a population genetic model by which humanity has a unique origin, that it starts from the first couple, Adam and Eve. But before I continue, I should say that this is ongoing work, so it's a proposed model which hasn't been validated with real data yet. And this project is actually started 5 years ago when I met Anne Gauger, who is a biologist at the Biologic Institute. And Anne, she had been looking at a region of the human genome that is very important for the immune system, the HLA region. And it had been, the argument was that this region is genetically so variable that it's not possible that humanity started from the first couple, especially when you look at this particular region. But she had also found some interesting articles that made it plausible that in fact there was a first human couple, and she wanted to continue with these ideas, and she wanted to have cooperation with a population geneticist, and that is how I got involved. And later on also Colin Reeves, who is a mathematician from Coventry University, joined the project. And last year we published 2 papers on this subject in Biocomplexity, and I will sort of describe now the basic features and the background for these population genetics algorithms for the origin of Homo sapiens. So let me give an outline of the talk. I will sort of start to contrast the sort of 2 competing human history models. The traditional one in academia is Darwinian macroevolution, and according to that model, we share common descent with the chimps. According to this model, the species alive today, which is most closely related to humans. And the competitor is unique origin models of humans, where we start from the first couple. And in order to sort of compare these models, we need to look at DNA variation among humans. And that is actually sufficient if we only want to make it plausible that a unique origin model of humans, to make such a model plausible, that is to validate it with data. It's sufficient to validate it with DNA variation among humans. And also if we want to compare it with a common descent model with chimps. But it's also possible to bring in DNA variation between humans and chimps and within chimps. And then, but in order to use this data, DNA data, and to say something about human history, we need population genetics. And there are a number of mechanisms of genetic change that population geneticists use that sort of explain why the genetic makeup of a population changes over time. And I will describe them, and also how that can be used to infer the human history. And I will also then describe very briefly two possible unique origin algorithms, where you either infer human history by simulating forwards in time or backwards in time. So the algorithms are simulation based for inferring human history. So let's start by contrasting these two competing models. The common descent models, according to that, the human and chimpanzee ancestral lineages, they split 6 million years ago here. And along the human lineage, Homo erectus evolved in Africa, and it spread to other continents about 2 million years ago. And they eventually evolved to humans and Neanderthals. But within these models, there are two versions actually. Something happened here. Now let's see. Yes, sorry. Either we have multi-regional or out of Africa. According to the multi-regional hypothesis, humans arose from Homo erectus at several continents in parallel. So when Homo erectus spread from Africa 2 million years ago, humans evolved in parallel at several continents. Whereas the more popular model, which is the out of Africa model, it says that humans arose only from those that part of the Homo erectus group that stayed in Africa, and that happened much more recently. And about 50,000 years ago, when Homo erectus had evolved into humans, emigrated from Africa to other continents, to Europe, Asia, and America, and so on. But there is also a hybrid model, because recently it has been possible to type Neanderthal DNA and other species, and that data strongly suggests that there has been interbreeding between humans and Neanderthals, and since Neanderthals are thought to be descendants of early spread of Homo erectus from Africa here, whereas most humans came out of Africa here, But if there has been interbreeding, there is sort of that gives rise to a hybrid model where most humans descend from Homo erectus emigrating from Africa, but there is also some interbreeding with Neanderthals that originate from earlier dispersals out of Africa. So that hybrid theory is perhaps the most popular now. And the other competing model is the unique origin model, where humans descend from one single man and one woman, and this couple had no further ancestors. And I will actually present two versions of it. Either this first couple lived in Africa, and that typically gives an older age of humanity. And it is very similar to the out of Africa model, where descendants of the first couple emigrated out of Africa to Middle East, and from Middle East to other continents, Europe, Asia, America, and so on. The difference is that humanity starts from a first couple here in Africa, rather than evolving from Homo erectus. The other version of the unique origin model is the Middle East, and is the first couple originating from the Middle East, and that gives a younger age of humanity. And the difference between these two models is the direction of this arrow. Not in that, rather than the first couple starting in Africa, it starts in Middle East, and from Middle East there was dispersal into Africa rather than the other way around, whereas dispersal from the Middle East to other continents could be much the same for both models. But it typically gives a younger age of humanity, this Middle East unique origin model. So we want to compare the common descent and unique origin model in light of genetics. And as I said before, in order to do that, we need to look at DNA differences among humans. And we could possibly also, if we want to do a more elaborate analysis, also include DNA differences between humans and chimps and within chimps. So let me start with DNA variation among humans. So here is a picture of nuclear DNA of humans that we have in all our cells. And the human genome, in the nuclear DNA, we have 22 non-sex chromosomes in 2 copies of each one. Females have 2 X chromosomes, males have 1 X and 1 Y. And together, all these chromosomes consist of 3 billion nucleotides written in a 4-letter alphabet, A, C, G, and T. And in the cell plasma, we also have a number of mitochondria with its own DNA. It's much shorter, only 16,000 nucleotides. And let's take, this is chromosome 1, for instance, in 2 copies of it from the father, inherited from the father and the mother. Let's take a very short part of that chromosome that consists of 14 nucleotides. We see that all the chromosomes, they form double-stranded strings that carry the same information. So because in each pair, the C always pairs with a G and the T always with an A. So it's sufficient to look at one of the 2 strands. And usually one talks about the coding strand, the one that most often codes for proteins, for those parts of the genome that are resided within genes. And the other is the non-coding strand. But individuals have different DNA strings. So these 3 billion nucleotides, that string is slightly different between different humans. That is what makes us genetically different. And there is actually different kinds of variation between humans. But the most common type of variation is variation at single nucleotides. And variation is called a polymorphism, so that gives us the name single nucleotide polymorphism. So suppose that we have two persons here. For this region of chromosome 1, these 14 nucleotides, the first person has a T in this position, whereas the second one has a C in that position. Otherwise, the two coding strands are identical. So then this is a single nucleotide polymorphism with two possible variants, two alleles. And most SNPs actually have two alleles or variants. And in this case, suppose that if we look at the worldwide human populations, 60% of all chromosome 1 copies of this region have the T, and 40% have the T and 60% have the C. Then we say that 40% is a minor allele frequency, because 40% is less than 60. And this we could do for all SNPs, all single nucleotide polymorphisms. And there are several million of them, and that's a lot, but remember that there are 3 billion nucleotides. So if we compare to humans randomly chosen and their DNA, there is on average a 0.08% difference, and that's called the nucleotide diversity. So that gives sort of an idea of how genetically different we are. And then one could do the same within chimps and compute nucleotide diversity. It's slightly higher within chimps, 0.13%. And then the next step is to compare the genomes of humans and chimps. And they are very similar in many aspects. You can sort of pair the different chromosomes, or it's only that the chimpanzee the genome has one more chromosome, but the human chromosome 2 corresponds to two chimpanzee chromosomes, 2A and 2B, that are shorter. The chimpanzee Y chromosome is shorter than the human, roughly half in size. But the question is, how big is the difference? And when the chimpanzee genome was sequenced in 2005, a few years after the human genome was sequenced, it was thought that the genetic difference was about 1%. But that mostly included single nucleotide polymorphisms. If one also includes other types of genetic variations, insertions and deletions and other kinds of things, the difference is probably quite a lot higher, perhaps something like 5%. Still, that's quite a small number, but the differences among those 5% is still very important, because humans have genes that are unique, that are not present among in the chimpanzee genome and so on. But that's a topic for a talk of its own. So here I sort of will more talk about the statistical population genetic aspects. So then, having now sort of looked at the DNA among humans and between humans and chimps, the next step is to summarize DNA variation in some convenient way. Because for humans, 3 billion nucleotides, that's a lot. And even though the SNPs are sort of a few million, that's also a very large number. So in order to get something more intuitive, we have to sort of extract some features. because we have something very high dimensional, we have to extract it to something more low dimensional. So we have to find some statistics that summarize the DNA variation. And I've already mentioned the first one. That is actually nucleotide diversity. So if we pick 2 humans randomly, there is a 0.08% difference between humans, and within chimps it's a 0.13% difference, and between humans and chimps, as I said, it depends how one counts, but it could be of the order of 5%. And the thing is that nucleotide diversity has something to say about the history of humans and of chimps and whether they have a joint history or not. So generally, if a population, that could be a species, if the nucleotide diversity is high, that could signify a large population, whereas a small population has a lower nucleotide diversity, because in a small population it's much more easy that alleles or variants get lost. And it has been thought that, well, for a young population, the nucleotide diversity has to be small. And that has been sort of the main argument against a first unique couple. 0.08%, it's a small number, but it has been argued that nucleotide diversity is much too high in order to allow for a young population with the first founding couple. But is it that way? Well, in this model, we sort of claim that that's not the case. A low population, a young population can still have a high nucleotide diversity if we regard this number as high. I will come back to the reason for this. But there are other more sophisticated summary statistics of DNA, because nucleotide diversity is just one number. We can surely extract more features from a string with several million SNPs. And the next summary statistics is the allele frequency spectrum. And in order to explain it, let me go back to this picture we had here, where I said that for each SNP with 2 possible alleles, we can compute the minor allele frequency. And in this case, it was a frequency of T. That was the minor allele frequency. That was 40%. And I said that there are several million SNPs, still a lot smaller than all 3 billion nucleotides, but we still have, there are several million nucleotides with variation or polymorphism. And for each of these several million SNPs, it is possible to compute the minor allele frequencies. And then one can sort of plot all these several million minor allele frequencies in a histogram. And that is called the allele frequency spectrum. So it's here. So this is the allele frequency spectrum. And that sort of gives some information about human history, because for an old population, the allele frequency spectrum has many more rare alleles, whereas it is flatter for a young population. And one can also sort of infer other aspects of the history of the population as well, but these are 2 examples. So that was the second summary statistic. The third summary statistic is the block length of DNA. And it turns out that DNA variation is sort of, for instance, within humans, is very much confined to block variation. So there is very little variation within the blocks. And these blocks vary in length, but their length is of the order 10,000 nucleotides. And so suppose now that we look at a portion of chromosome 1. Individual 1 has 2 copies of chromosome 1 from the father and mother, and individual 2 has 2 copies as well of chromosome 1 from the father and mother. And at this block, there are 4 versions of this block. So the first individual has the yellow variant from the father, the green from the mother, and so on at the second block and third block, and so on. And now it turns out that this block structure also gives some information about the history of the population, because a young population typically has longer blocks. The block length is larger, whereas an old population has smaller blocks. The block length is smaller. And why is that? Well, that has to do with something called recombinations in genetics. When a parent, a mother, for instance, transmits transmits DNA to a child, she either transmits the grandmother's or the grandfather's DNA. And there are, along all non-sex chromosomes, there are switching points between grandmaternal and grandpaternal mode of transmission, and those are the points of recombination. And really, the boundaries between these blocks are ancestral recombination events between grandpaternal and grandmaternal mode of transmission that happened a long time ago. And it's natural for a young population, there haven't been that many ancestral recombination events. So the mosaic is coarser, whereas for an old population, there have been many ancestral recombination events. So there is a finer mosaic of blocks. So we have 3 kinds of summary statistics that all of them give some idea about the history of the population. But how do we get from the summary statistics to draw these conclusions here about the history? And from allele frequency spectrum, we draw some conclusions about the history, and from the block length as well. Well, that's where population genetics comes in. Because population genetics uses mathematics in order to describe how the genetic composition of a population changes over time. And using that in order to infer the most likely history from this DNA variation that we see, that we sort of have summarized using these statistics. And this history will sort of look different depending on what kind of DNA we have. If we look at Y chromosomes, only males have the Y chromosomes, so they are only inherited from fathers to sons. So if we sort of look at 3 males here, so their ancestral tree will be a male tree. So suppose, for instance, that males number 2 and 3, if we go maybe several hundred generations back, their lineages merge or coalesce. Then if we go a number of generations further back in time, it coalesces with the ancestral lineage of the first man. And that is a male ancestral tree. If we look at mitochondrial DNA, which is passed on from the mother, we get similarly a female ancestral tree. It's more complicated if we look at ancestral trees of non-sex chromosomes and X chromosomes, because it's transmitted both from males and females. And this actually implies that if we look, for instance, at chromosome 1, the first non-sex chromosomes, at one part of chromosome 1, if we have 3 individuals here, they could be men or women, their ancestral tree will look different at different parts of chromosome 1. And the reason is that there have been ancestral recombination events in the past. Along the non-sex chromosomes and along the X chromosome, we have different ancestral trees when we look at different parts of those chromosomes. But it's those kinds of histories that we sort of infer from a population genetic study. But in order to do that, we need to describe these mechanisms that are used in population genetics, these mechanisms of change. And there are 5 of them that are usually used in population genetics to explain how the genetic makeup of a population changes over time. And the first one are mutations in germ cells, and they happen very rarely. About the probability is 10 to minus 8 per nucleotide per generation that a mutation happens. And let me just explain here. Here, when a germ, this is part of a, let's say this is part of a sperm cell when it is formed, a 14 nucleotides long string, double-stranded. And suppose now that when this sperm cell is formed, there is a change in the coding strand from a C to a T. And then when this sperm cell then unites with an ova cell and we have fertilization, this mutation will spread to the child, to all copies of that cell. And it's only these kinds of germline mutations that are of interest in population genetics. The second kind of mechanism of change is genetic drift, and that is changing the proportion of different variants that are already in place. Mutations create new variants, new alleles, but genetic drift only changes the proportions of alleles that are already in the population. So I will explain this with a male population with 10 individuals in each generation. We have 5 generations. So suppose now that in the first generation, this male number 8 has a T in that position. All the others have a C. And then it happens that this male gets two sons. And then that means that the frequency of T of this allele will increase from 10% to 20% between generation 1 and 2. And then in the third generation, these 2 sons altogether have 3 sons in the next generation. So then the frequency of T increases to 30%, but it sort of oscillates up and down due to chance, depending on how many children those men with a T happen to have. And sooner or later, one of the 2 variants will be lost. And this happens much more quickly in a small population, because then it's sort of easier by chance that either a T or the C is lost. So that means that genetic drift reduces genetic variation, whereas mutations creates new variation. Selection is very similar to genetic drift. The difference is only that some alleles tend to have more, individuals with some alleles tend to be more fit. So on the average, they have more offspring. So let's go back to the same example. We have this population of 10 men in each generation. And suppose now that men that carry a T in their Y chromosome are more fit. So T has a selective advantage. So those men tend to have more children. Well, what happens is then that the frequency of T will increase more quickly, and quite soon it will sort of spread to the whole population and get fixed. That means that C disappeared and there is only T left in the population. And there are several types of selection, but this is called directional selection. And it has the same effect as genetic drift. It reduces variation, and that happens more quickly than for genetic drift, because then the frequency of the alleles oscillates up and down. But if you have selection, there is more a systematic increase of one of the 2 alleles until the other gets lost. Recombination, I mentioned that a little bit before. That is switching between grandmaternal and paternal mode of transmission. So let me illustrate this with, let's say that this is chromosome 1 for a male. One copy inherited from, so this is the father. So the yellow chromosome inherited from the grandfather and the pink one from the grandmother. When here 2 sperm cells are formed, and in the first sperm cells, the first half of the chromosome 1 copy is inherited from the grandfather and the second half from the grandmother. And in the second sperm cell, it's the other way around. And that means that combinations of alleles at different positions are recombined, and that's where the name comes from. Here is a point of recombination, but whereas in the father, a G was always combined with a T and an A with a C, in the 2 sperm cells, a G is combined with a C and an A with a T. So we see that recombination it doesn't generate new genetic variation, but it sort of reshuffles variation that is already in place, and it creates new combinations of alleles at different positions. So that was the 4th mechanism of change, and the 5th one is then we look at a species or a population that is sort of spreads over a large region, and then there is sort of colonization. So people sort of this population is fragmented into different groups, and there could be some migration between these subgroups, and that gives sort of regional variation between the DNA makeup of the different groups or regions. And let me illustrate. Let's go back to this population of 10 males and their Y chromosomes. Suppose that 4 of them come from Europe and 6 from Africa. Then all of these 10 males get one son. So I have depicted the son right below their fathers, except for this father, his son moves to Africa, and this first African father, his son moves to Europe. And we see that whereas in the first generation, there is a big difference in the genetic makeup between Europe and Africa at this nucleotide. All the Europeans have a C and all the Africans have a T. Because of this migration, this regional change between Europe and Africa, it sort of decreases, because now there is a T among the Europeans and a C among the Africans. And similarly at this second nucleotide as well. Whereas colonization or isolation between regions has the opposite effect, that will increase regional genetic differences. So these were the 5 mechanisms of genetic change that population geneticists use to sort of explain how the genetic makeup of a population changes over time and to infer history. And based on, we see that I've only kept the first mechanism, mutations, because that is the only one of these 5 mechanisms that sort of create new diversity. Genetic drift and selection, they remove diversity, whereas recombination and colonization migration, they sort of in a way reshuffles variation geographically or along the genome. And it has been said that, well, it's not possible to have a human history model with the first human couple, because there is too much variation within humanity, 0.08% nucleotide diversity, because there's only one mechanism that creates new diversity, and these are mutations. And they happen so rarely that humanity cannot be young, and it's very difficult to have a first couple unique origin model. But the main idea, now comes the main idea, and it's actually not our idea. Others, John Sanford and Robert Carter and Nathaniel Jansen, they also worked on models where we have a sixth mechanism that I also kept, because if there was a first couple, it could have been created with diversity from the start. And then we get a sixth mechanism of change that also generates diversity. And this is possible for non-sex chromosomes, because in the first generation, if Adam and Eve existed, Adam had 2 copies of a non-sex chromosome, Eve had 2 copies, altogether 4 copies, and those 4 copies could have been created differently. For X chromosomes, Eve had 2 copies and Adam 1 copy. That gives a total of 3 copies. They could have been created differently. For mitochondrial DNA, it is Eve that passes on her mitochondria to her daughters, and she has lots of mitochondria in the cell plasma, and they could have been different as well. It's only for Y chromosomes that this doesn't work, but on the other hand, the diversity is less for Y chromosomes than for other types of DNA. So let me illustrate this. This is the first couple, Adam, Eve. Here is one of their children. Here we have three nucleotides, which are all polymorphisms. There are variations at the blue nucleotide, at the black, and at the red one. But this diversity is created at the blue and red nucleotide, because this child received the T from Adam here and the A from Eve. And the same for the red nucleotide. The child received the C from Adam and the G from Eve. Whereas here we also have a polymorphism, but it was not created because there were only 4 Gs in the first couple, but there was a germline mutation taking place when the ovum cell was created. And if now the first human couple is fairly recent, then it's likely that created diversity, according to a unique origin of humanity model, that created diversity accounts for most of the variation, and much more than germline mutations, because there hasn't been enough time for germline mutations to accumulate. So that was a 6th and very important mechanism for a unique origin model, to sort of include that mechanism in the model. So now the next step now is to use these mechanisms and try to simulate and validate different histories, and in particular to compare the unique origin and the common descent models. So first we need to propose a history of humans and chimps, and it's not enough to say that we need to have a common descent model and we need to have a unique origin model. If we study the unique origin model, we have to specify a large number of parameters. When did the first couple live? How did the population size vary from the first couple until today? Was it split into different regions, and when did that happen, and so on? What was the generation time? And the ratio between males and females, was it almost always close to 50%, and so on? And that we have to specify for both common descent and unique origin models. Given that we have sort of come up with a history, we now simulate genetic data from it a large number of times. And then we sort of want to summarize that simulated data with those summary statistics. So suppose we simulate genetic data for humans and chimps 1,000 times. Then we sort of get 1,000 summary statistics. And here I have sort of depicted how this could be done in principle. If we have the common descent model, we start with a DNA of a common ancestor of humans and chimps, and then that DNA is spread during several million years to DNA of humans and DNA of chimps, and then we need to summarize the DNA of the 3 billion letters along the human genome into those summary statistics I mentioned: nucleotide diversity, allele frequency spectra, and block length. And we do the same for the chimp DNA. So here are the summary statistics. If we now simulate this 1,000 times, we get 1,000 copies of these summary statistics of DNA. And then we do the same for the unique origin model. But then we do this separately for humans and chimps. So for humans, for instance, then we have the DNA of the first couple, and then that DNA is spread to DNA of humans of today and summarized using these different summary statistics. And we see that if we have the unique origin model, we have created diversity here. And then all these other 5 mechanisms: mutations, genetic drift, selection, recombination, and this is colonization, migration, they sort of act on the way from the first couple down here. Whereas for the common descent model, we don't include created diversity, but only the other 5 mechanisms of change. So if we repeat this, let's, for instance, look at the unique origin model for humanity. We have this summary statistic, for instance, nucleotide diversity. We repeat the simulation 1,000 times. So then we get 1,000 simulated nucleotide diversities. And we plot those simulated nucleotide diversities in a histogram and compare it with observed data, and that was 0.08%. And if the proposed history is good, the simulated copies of the nucleotide diversity should be centered around the true value. But if the histogram is down here, that's a bad fit. And then we do the same for the allele frequency spectra and the block lengths. The simulated copies should center around the observed true values. And of course, it's a huge parameter space to propose different kinds of histories. As I said, there are a number of parameters to vary. So a good approach is to start with some very crude models for the history and then refine it successively. So that was in principle how one is doing. So now let me go back and to ask the more rhetoric question: why should we try a unique origin model at all? Because so far it's only common descent models that have been validated with real data. And as I said, it's only the 5 first mechanisms of genetic change that have been used, not created diversity. So shouldn't we be satisfied with the common descent models? I argue that unique origin models should be validated as well. I mean, as a Christian, I could argue that from the Bible, I read that Adam and Eve, they were created unique, and I would like to try a model that sort of makes it possible to have a more literal interpretation of Genesis. I think that's a good argument, but there are a number of arguments that one can use on top of that. So although I think that's a good argument to try different things from the Bible, we don't need that argument, because there are a number of other arguments why it is of interest to try a unique origin model. First of all, created diversity, in my view, possibly makes it a strong competitor to the common descent models in order to fit data well. So that is the first reason. And then secondly, I think a unique origin model seems very consistent with the observed block structure. It's also called haplotype block structure. Remember, I said that DNA variation is along blocks. It's like we have different mosaics of those blocks of length 10,000 nucleotides or so. And the interesting thing is that each of those blocks come in, let's say, 3, 4, or 5 versions, but approximately 4 versions. And that seems, if we look at non-sex chromosomes, if there was a first couple, Adam and Eve, there were 4 copies of each non-sex chromosome, 2 from Adam and 2 from Eve. And then it's very consistent then to have that kind of haplotype block structure, because then it's simply so that we have different mosaics of the 4 chromosomes from Adam and Eve, and the boundaries between those blocks are old ancestral recombination events. So that is the second reason. The third reason is that a unique origin model, in particular with a young age of humanity, faces less problems with inbreeding depression, because it is actually the case that when each new baby is formed, although mutations happen very rarely, remember that we have 3 billion nucleotides, each one of us, in our genome, and the probability is 10 to minus 8 at each nucleotide to have a mutation. So there are at least 100 new mutations in each newly born child that have never been seen before. And most mutations are neutral or slightly deleterious. And that means that those deleterious mutations, they tend to accumulate over time. And John Sanford has written a book about this. And natural selection has difficulties in removing all those slightly deleterious mutations, and that could, in the long run, create problems with inbreeding depression, when sort of those harmful variants, when some children get 2 copies of them from the father and the mother, and but that problem that's the smaller problem if humanity is sort of younger. So that is the third reason. And the fourth reason might be that interbreeding with Neanderthals becomes less problematic if we simply assume that Neanderthals were humans and descendants of the first couple. With the currently dominating view is that Neanderthals and humans split at least 500,000 years ago, and then they sort of there was interbreeding much more recently, 50,000 years or even more recently, and that could be very challenging. That 2 sort of species that have been separated for such a long time are able to get fertile offspring. That could be another reason why it's of interest to look at unique origin models. And before I close, I would just like to say that how one could possibly simulate from a unique origin model. That this could be done forwards in time or backwards in time. And others, John Sanford and his group has worked with a formal simulation approach. So the novelty in our work is really the backward simulation. So let's first go back to a picture I had before here. So what I'm talking about now is a unique origin model, and how do we simulate DNA of the first couple down to the present generation in order to then summarize those statistics. So this could be done in 2 ways, either forwards in time or backwards in time. So let's go back to the forward simulation. And that is, suppose we want to simulate DNA of 2 individuals from the current generation. And for the forward simulation approach is very general, but is quite slow. because that means that we first, according to the history we have picked from the beginning, we start with the first couple, Adam and Eve, generate their DNA with created diversity, and then we sort of stepwise for each generation build the ancestral tree forwards in time and spread DNA along with that until we come to the parental generation. But we have to do this for the worldwide human population. So it's a very large ancestral tree that we have to build. So it's computationally quite slow. And then from the parental generation, we simulate DNA for the two individuals that we sort of wanted in the first place to simulate DNA from. On the other hand, this approach is very general. We can build in various types of selection and other genetic parameters into this model. The other approach is backward simulation, and it's not as general, but it's faster. So that's sort of our main contribution, that we and this is a well-known technique in population genetics. It's called coalescence theory. So we sort of apply this technique in this unique origin model framework. And the goal is the same. Suppose we want to simulate DNA for two individuals from the current generation. And now we do it in the other order. We start with these two individuals, and then we build the ancestral tree backwards in time until we reach the founder generation. And that tree is a lot smaller because we only have to include those chromosome copies that have passed on genetic material to the current generation. And that tree is a lot smaller than in forward simulation. And then, so that is the first step, building the ancestral tree backwards in time. And when we reach the founder generation, we sort of go the other way around and spread the first couple's DNA along this much smaller ancestral tree forwards in time until we reach the current generation. And then we have simulated its DNA. And so that sort of completes my talk, and you can find more details in those two papers we published last year in Biocomplexity. The first paper is shorter. It gives a qualitative comparison between common descent and unique origin approaches, and in the second paper is longer and more mathematical. There we sort of present this unique origin algorithm based on backward simulation, and there are lots of further references in these two papers. So I want to thank you for your attention, and I would be happy to take questions. Thank you.
Well, thank you, Dr. Hössjer. That was a very fascinating presentation. Do you want to unshare your screen at this point?
Yes. Stop sharing.
Yep. Great. Folks, this is the interactive portion of the program. If you would like to ask a question, you can submit it in textual form by hitting the Q&A button at the top of your screen and typing it in. Or you can also click raise hand, in which case I'll promote you to be a co-panelist, and you'll have opportunity to go back and forth and discuss what we've been talking about. We have a couple of submitted questions already. Alistair says, these mechanisms of change in the way you've described it, inherited from grandparents, don't seem to affect the viability of the organism, yet in other contexts, variations of just one nucleotide has catastrophic consequences to proteins because it disrupts binding sites of proteins. What is being varied and how does it express itself without being a lethal change?
These are sort of genetic changes, and they will sort of then from because of the genetic code, if we have genetic changes, they will sort of it depends on what kind of mutations we have, but some genetic changes will affect what transcription of proteins, and that will sort of affect the machinery in the cells. So that's a good point, that it is connected to the fact that some mutations are neutral and others have a selective advantage and so on. But I could say that selection is always on the phenotype, different features like body length, muscle strength, and things like that, rather than on genotype. So that's important to bear in mind as well. So I don't know if that answered the question, but
cool. Jacob Guthrie asked, does the unique origins model fit all the data we have from population genetics?
yeah, that's a very good question. Well, I mean, we haven't really validated with real data, but we tried to make this model in the second published paper as general as we could. So we wanted this model to include various types of genetic data from Y chromosomes, X chromosomes, mitochondrial DNA, and non-sex chromosomes, and also sort of incorporate different kinds of histories. The problem is that on one hand we want to make it general, on the other hand it's very difficult to fit a model with too many parameters, because it's a sort of a huge parameter space. So it's always that balance. But in principle, we believe that the model includes many features. But still, in order to make it sort of practical, at least to begin with, we have to start with much smaller models. So and sort of learn from experience. So yeah.
We had a hand raised from Dr. Therese Nortney. Therese, are you on?
Yes, can you hear me?
Yes, we can.
Yeah, hello, Therese.
Hi, how are you?
Fine.
That was a really great talk, and I'm glad that you went through a lot of the basics. Because this is not really, I don't know, like a lot of people have like a general idea of how evolution is thought to work, but when you get down to all this terminology and step by step what is actually changing over time and what are those mechanisms, it's really clarifying.
Thank you.
Yeah, so I appreciate that. So I'm like sort of vaguely familiar with you and Anne, what's the other? I know him. Anyway, your other co-author did, but I have been thinking a little bit about do you feel it's possible to tell the difference between the diversity that was created originally and the diversity that showed up after that point?
Yeah, I think that's a very good question. But I think if we look at this haplotype block structure, it's like that. It's like that we have, let's say that we have 4 copies of each block, and then the original blocks, they comprise the created diversity. And then within all these 4 copies of a block, we have a little within block variation as well, and they are due to the germline mutations. So if it would be possible to sort of start with this block structure, and then say that the original variation between the copies, the 4 copies of each block, are due to created diversity, and then the finer variation within each of these 4 copies are due to germline mutation. So maybe through this block way of thinking, it could possibly be done, but I haven't really thought about it in detail. But that is my intuitive feeling, that it could possibly be done in that way. And I guess it depends a lot also on how old humanity is, because if as I said, if humanity is very young, then the created diversity will dominate. Whereas if it's older, then the germline mutation has a big role to play, so to speak.
So do you feel like this is a situation, before that I should ask, what type of data would you need to validate your model?
Yeah, that's also a very good question. I mean, One could start with DNA just from one region to make it simpler, to just look at DNA from among Europeans or some other region. But to really make it more general, it's very important to have DNA from people all over the world, because we need to capture these different dispersal patterns that we talked about. If we have a unique origin model that starts from the Middle East or from Africa, then it's very important to have DNA and different types of DNA as well from different regions, from the Middle East, Africa, Europe, Asia, and so on. So, but that also makes the model more complicated, because in the history we have to build in scenarios as to when did the descendants of the first couple start to move geographically, and when was the population split into geographic subunits and so on. But the answer is that we need DNA, different types of DNA, and DNA from different regions all over the world. So, yeah.
So, like, in theory, is this not quite like disprovable? Like, so what I'm wondering is, given the access we have to, even if we could sequence everybody's genome who's living right now,
Yeah.
do you feel that there are too many unknowns and too few equations to be able to differentiate between which model fits the data better?
Yeah, that's a very good point. And that's a major problem with population genetics, that it is very difficult to infer history with certainty, because there are so many scenarios that give almost the same output.
Mm-hmm.
But I think still today we have sort of much more genetic data, so it's at least easier now than like 10, 15 years ago. So it could be that in the limit we can sort of say that we cannot distinguish those histories from each other from genetic data alone, but maybe we can sort of distinguish certain groups of histories from each other, and it could be, yeah, and things like that.
So, like, when it comes to like X chromosome and mitochondrial, or sorry, Y chromosome and mitochondrial DNA, wouldn't the story be sort of different as far as the diverse? I mean, so there would be one original copy that is going to be the progenitor, or should I say ancestor, or the ancestor of all of the copies we have now. So the idea of creative diversity there doesn't quite hold as much, right? So would that be a way of testing, or did you guys look at that aspect in your model?
Yeah, that's an excellent point. Actually, it's only for Y chromosomes that we only have one version in the first couple, because Adam has one Y chromosome and Eve has not. But for X chromosome, we have the creative diversity there as well.
I mean, mitochondrial DNA, sorry.
Yeah, mitochondrial DNA, yeah. I guess you can argue in both ways. Because Eve has several mitochondria in her cell plasma, so they could have been different as well. So maybe we could argue we created diversity in mitochondrial DNA as well. But surely for non-sex chromosomes and X chromosomes. But in any case, you're completely right. We cannot have that argument for Y chromosomes. Yes, then we need to stick with the other 5 mechanisms of genetic change. But it seems that from data that the genetic variation among humans, for instance, in the Y chromosome is a lot smaller than for other types of DNA. And that's very interesting, because that would then, without having sort of really validated and fit data, that could be sort of a preliminary fact that sort of makes the unique origin model promising. Because otherwise, I mean, Y chromosome data is a big challenge for a unique origin model, because we cannot include the creative diversity. But on the other hand, the variation among humans, among men, in their Y chromosome is a lot smaller than for other types of DNA. So that sort of fits well with the unique origin framework. But that's a key point that you mentioned, and that's why it's important to have all different kinds of DNA included in the model.
So do you feel like you've set up a way of arguing from the data and not from an axiom of let's let go of common descent? Like, I think it is valid to say we should be agnostic about common descent, not married to it, right? As far as like that chimps and humans had a common ancestor. But do you think there's a way in the techniques and the models that, in the model building techniques that you guys have generated here, to show that it's a plausible inference from the data that common ancestry is a poor fit to what we see?
Yeah, I think what we have proposed is sort of because so far only common ancestry models have been fit, and because there, I would say that because there are so many parameters to vary in a population genetic model, even though, I mean, either the unique origin model or class of models is correct, or the common ancestry class of models is correct. But within both of these classes, you have an enormous amount of parameters to vary. Mm-hmm. So even if you start in the wrong class, you may end up fitting data quite well. Right. And that's also an argument, because people can say, now we have fitted common descent models and they fit data rather well. Yes, but you have an enormous amount of parameters to sort of vary, and it's well known that in population genetics it is hard to distinguish various histories. So for that reason, it is very important then to try the other class as well, because there we also have an enormous amount of parameters to vary. So it could very well be in the end that both models fit data well, and then the outcome in, well, we don't have to abandon unique origin because of genetics, although but I also argued, I had some other arguments why I prefer unique origin, but it may still be that both models fit these summary statistics quite well in the end, because there are so many parameters to vary within both classes.
Yeah, and I would even say, the way I would look at that, I think that's a really great outcome, honestly. If given the number of parameters you can vary, and anybody who's ever tried to fit something knows that if you have enough variables, you can overfit, your data, and really you can end up not having anything valid at the end. And so the idea that that's just the reality of what these historical models, the reality of the problem these models present, would say not that we don't have to abandon the unique ancestry model, but that there's no reason to conclude the common history one.
Yeah, exactly.
It's not that we have something we don't want to let go of. It's like we have two things that we don't have a reason to conclude one or the other, and that's what the science is telling us. So if somebody wants to say that one is scientific and one is not, actually the science says that you can't say that about either. Like, the science says you have to take them both and consider them both until you have additional data or additional arguments or whatever to clarify. So I think that's a really powerful conclusion.
Yeah, that would be great. Yeah, I agree. Because then if people say that, well, we cannot have as you said, a unique origin model because genetics has disproved it, then if the outcome is that, well, your unique origin model explains data as well, then that would be great. And so yes, I agree. Yeah.
Cool. Well, if anybody else wants to get on, I can stop telling you.
There are a few submitted questions, and perhaps go to them.
Thank you for the question.
No, who's a Muslim apologist, says, what is the difference in evidence between the out of Africa theory and the out of Middle East theory? I'm just wondering, is this entirely based on archaeological evidence, or is there some theoretical genetics involved here?
Yeah, that's an extremely good question, because both of them have their pros and cons. I mean, if you want a literal interpretation of Genesis, then that is closer to the Middle East unique origin model. But if we only look at data, the reason is that if you look at data, African DNA looks older than for the rest of the world, and all other populations are quite closely related. So that's the reason for the out of Africa hypothesis, that humanity started in Africa, and then from there 50,000 years ago they dispersed to Middle East and then to the rest of the world. So as I said, the Middle East ancestry model, then the direction goes from the Middle East into Africa and then to the rest of the world. So an advantage of the Middle East origin approach is related to these questions I said about inbreeding depression, because as related to John Sanford's book, if humanity, because Middle East origin is coupled to a young age of humanity, and then there has been less time for inbreeding depression to accumulate over time, and also this question about interbreeding with Neanderthals and so on, it sort of is perhaps also is easier within the Middle East ancestry model, and as well this haplotype block structure, which seems consistent with the fairly young origin of humanity. But on the other hand, what sort of the difficulty of the Middle East and younger dating of the first couple is that African DNA does look older, and that would sort of then one would think, well, then the first couple should be placed in Africa, and that one could see from allele frequency spectra, nucleotide diversity, and block length that African DNA looks older. But it looks older, it is not necessarily so that it is older, because there are tentative explanations that we mentioned about in the paper, but that these are things that have to be explored more. It could be that there is a lot more tribe isolation in Africa. You can see that from languages and so on. And if you look at DNA for a population that is very divided into small subunits that have been isolated for a long time, then their DNA, if you look at the DNA variation of the whole region that consists of those very tiny tribes or subunits, then it looks older than it is. So that could still be a possible explanation. So both models have their pros and cons, but it's an excellent question. And that is sort of, so the first thing is to sort of choose between common descent and unique origin model. And then the next step is to choose between the two, given that one of the two unique origin models is fairly competitive or better. The next step is to choose between those two.
Anne Gager, your co-author in the Q&A submission button, she said, good point, Therese, that is actually the point, that if we have two models that both fit the data equally well, the population genetics model cannot claim theirs as the only possible answer.
Yeah, that's an excellent point. Yes. So if that is the outcome of this project, we are very satisfied. And if the unique origin model works better, then I'm even more satisfied. But if I'm very satisfied if this is the outcome. Yes.
I don't know if Anne wants to hop on the panel and contribute herself, but she's welcome to do so if she wants. Alistair submitted a question saying, what critical responses have you had from skeptical readers, and how have you responded so far?
Well, I think I've had some critical remarks about, in particular, when we compare these two unique origin models, Middle East versus Africa, and then we had some arguments that in order to explain African DNA looks older, but it doesn't need to be older. And then and then we refer to some papers in relation to that. And there we had some critique regarding to that. And it's still sort of, these are sort of tentative explanations that we mentioned, and some of them might be wrong. So but that is sort of a bit of fine calibration with choosing between the two unique origin models. But I think also that I think otherwise, because the two papers came out quite recently, so there hasn't been too much criticism either otherwise. But I think probably some people that more advocate evolutionary common descent model would think that if humanity started very early, very recently, the allele frequency spectrum will be too flat, because that is the case for a young population. But my hope is that it's still possible to have many rare variants if, as we know it is the case, that even though humanity started uniquely, if it started recently, and if the population size increased very rapidly in recent times, it's still possible to have many rare variants. That's my hope. That's still a question that has to be sort of looked at in more detail. But so those 2 points of criticism I've heard. Arguments why African DNA need not be older, although it looks older, and those objections could be true. And the other one is the allele frequency spectrum and many rare variants. Is that possible for a young origin model? Well, it could still be, I think, if the population growth was very quick the last sort of centuries or so.
All right. Anne Gager has joined the panel. Hello, Anne.
Oh, hello, Anne. Hello.
Hi, Ola.
Hi, hello.
One criticism I've heard is that the model is too comprehensive.
Yeah, and yeah, that's true, and that's a very good comment. There are too many parameters in the model, so the idea was really not to fit them all, but really start with a huge model, and then when we fit data, just to vary some parameters. But the thought was that when we describe the model, we include everything that possibly could be included. But then when you sort of fit data, you should just you should start with very crude models, and then successively add more parameters, sort of fine calibration.
Right.
Yeah.
Well, I think the person who made that criticism was in actuality overawed by it because it is such a comprehensive model.
Yeah, maybe. Yeah. And it is sort of that could be the case. Yeah, truly.
Yeah.
So I think we have to do this stepwise and sort of start with crude models. Maybe just start with one region to try to fit European DNA variation at first, just to learn more about the model and so on.
Right.
I have a question about some. I haven't read your whole paper yet, but what about sexual selection and variances in sexual selection over time, over human history, and across geographic regions? Like, how do people take that into account? Because when it comes to inferring the age of a population based on genetic variation, the assumption has to be made that all sexual selection functions are equivalent or the same across the whole population, but that doesn't seem even like an at all sound assumption to make to me. You know what I'm saying?
Yeah, and I don't know very much about the details here, but I could say that selection. I mentioned at the end that there were two versions how to simulate from the unique origin model: forwards in time and backwards in time. I said that our approach backwards in time was sort of quicker, faster, but it's not as general. So in order to simulate the ancestral tree backwards in time, we mostly have to assume neutral models. And since we deal with genome-wise statistics like allele frequency spectra, nucleotide diversity, even though there is selection in some parts of the genome, one could argue that most of the variants are fairly neutral. But one could, on the other hand, look at just a certain small region where there is known to be selection, and then the backward simulation approach is not maybe the best. Then, because if you use forward simulation, then you can build in all different kinds of selection. For instance, sexual selection, balancing selection, and all different kinds. And I think then you have a slower algorithm, but then you can build in those kinds of things. But my knowledge about sexual selection is too small in order to sort of know how much it accounts for the variability of human data. But it could, that is one interesting parameter to vary, surely, to see how important it is. So
there was a question submitted by Alistair, who said, one of the arguments that I've heard against unique origin of humans is the similarity of the second chromosome to the split chromosomes in chimps. Does this approach provide a response to that problem?
Yeah, I think Gauger knows more about this, but I mean, it's striking that it was thought to be just 1% difference between human and chimp DNA. Now it's more of the order 5%, my general feeling, I mean, not only talking about chromosome 2, but looking at the whole genomes of chimps and humans, I think, I think you are better answering this, but it's really the 5% differences that are important, because humans have several unique genes that the chimpanzees don't have, and the gene expression is very different. So even though 5% might at first sight look, it seems as a small number, but if you look at those 5%, they sort of correspond to very big differences in terms of genetic contents, and also the 95%. There we have differences in gene expression, especially in genes that sort of regulate activities in the brain or so. But this is really things that I've learned from Anne, so she should answer this. So what is your comment?
My comment on the second chromosome fusion is that there's no evidence to say that fusion happened before or after any posited split of the chimp and human lineage. In other words, the fused chromosome could have been original. Mm-hmm. And there's no indication time-wise that it had to be anything else. So in which case the argument against unique origin doesn't matter. Unique origin would start with a fused chromosome, or what appears to be a fused chromosome.
Right, because then we're just simply back to the argument from similarity, which distinguish descent from design in that case.
Right.
So, like, what kind of evidence would point to time-wise a fusion before the human split? Would it be part of the chimp population or part of the non-human primate population having two chromosomes there instead of
the evidence simply is that chimps have two and we have one, and there are similarities in our sequence. There we go.
And I mean, it could. I mean, one could develop an argument if we were able to find evidence pertaining to other hominid lineages that are not Homo sapiens, because then if the chromosome is also fused in that, it would suggest that on Ola's model, where basically you've got the created diversity situation, you would need to then postulate that Homo erectus and Neanderthals and Homo sapiens and all the other Homo species were descendants of that primordial pair, and that I think would lead into some significant problems. Anything to say to that, Ola?
Yeah, you mean yes. I think I mean anthropology is not my specialty, but as far as I know, I mean Neanderthals are very similar genetically, and far as I understand, even if you look at fossils data, so they could very well be humans and maybe Homo erectus as well. So, but otherwise, uh, but I guess what I've learned from fossils data, that some fossils are very human-like and others are very ape-like. But there is a big gap between those two classes, and either Homo erectus and Neanderthals are really humans, or they are different Homo species, and then we have a sort of, in that case, we would have a sort of first ancestor of all the Homo species, and then yeah. So there would be different possibilities within those scenarios, I think.
But do you think, Ola, as a Christian, it's necessary to postulate that the different species of our genus Homo are one single species? Do you think that's necessary to postulate that? Because could you not say that there are, I mean, from a theological point of view, would you say that it's possible to postulate different species of humans which are not, endowed with the imago Dei and so on?
May I answer that?
Yeah, go ahead.
Charisse and I have had this conversation about the different points in history where Adam and Eve might fall, either at the start of Homo erectus or the start of what's called modern humanity about 200,000 years ago, or at the start of the migration out of Africa, or the start of the appearance of culture and agriculture. Those are all possible starting points. My preference for an older age, which is my preference, is because I don't know why theologically God would go to the trouble of making creatures that look like us that aren't us. So what's the role for Homo erectus if he's not human? And what's the role for the Neanderthal if they're not human? Particularly Neanderthals, of course. And that's my question. Other people have different reasons for choosing different points of view.
Mm-hmm. What do you think, Ola?
Yeah, I think I mean, I was for many years Christian. I believed in macroevolutionary theory, so it's sort of I think I mean it's you can have different viewpoints on this and be a sincere Christian, definitely, but from my point of view, the first chapters of Genesis are much more harder to interpret if Adam and Eve were not created unique. So it would, for me, it would be much easier to interpret Scripture if in that case Homo erectus and Neanderthals were humans as well. But if data tells the other way, then we have to accept it. But it's always difficult with historic data because there are so many assumptions involved in the dating methods. We have the radiometric techniques, and they also have assumptions inbuilt. But definitely, I mean, I guess it's still, for me, it makes it's much. I feel much more comfortable with interpreting the Bible in this way. But if it's a case that the data would say otherwise, then we have to be open for that. But yeah, so that's sort of my question, but my answer. Yeah,
because obviously there are species of primate which are similar to us in many respects and also different from us in many respects. So simply the fact that an organism looks similar to us in certain respects doesn't, I think, mean that they are endowed with the image of God, which we Homo sapiens are. So I don't think it would necessarily pose a theological conundrum if indeed Homo erectus and the Neanderthals and so on were were a different species of or genus Homo. I don't think that would necessarily cause a theological problem.
Yeah, that makes sense. Yes, that's.
I'm having trouble following because, I mean, again, this is the species problem. So when the way that we have scientifically labeled these things as species and genus and things, like it doesn't compute for me because it's totally arbitrary, and like it creates it creates the problem of trying to figure out like whether they were the same or not by saying that they're not the same, and by this particular set of degrees of difference, and that's meaningful. So to me, like a species is the descendant of an originally created kind. So the question therefore is Homo erectus and a descendant of Adam and Eve? That's really the question. And I don't know what kinds of data you would need to answer that question. But one thing that I find convincing is when you have admixture. And I think you were talking about this, Olaf, earlier, and I wanted to clarify when you were talking about colonization and migration. And migration results in decrease in the differences because you've created admixture, right? Is that basically the technical terminology for when you're bringing SNP profiles from a previously isolated population and interbreeding them into another population, and you can see later on the signatures of those mixed in SNP profiles, right?
Yeah, that's a mixture, yeah.
Right, so to me, one thing I feel is very conspicuous in its absence is we don't have like admixture of chimpanzee. We don't even do that, right? But we do have admixture apparently of Neanderthal that we can detect, right? So that is positive evidence of having been part of a genetically interbreeding population. And to me, these sort of asymptotes of genetic mixing that you see similarity, but you see like this gap of the type of similarity that you can call it. So you go from having an allele to now it's a substitution. What's the difference? It's a change, but why did we create new terminology for that? Well, because it's a change across a species barrier, right? So to me, that's like screaming, this didn't come about necessarily. This didn't come about through descent with modification. And therefore, I feel it's helpful. I don't know, I don't know the answer to the like Homo erectus and all of that, but I think that for sure once you have evidence of having had, children together in the past, that you probably came from the same original parents, because that is just using the uniformitarian method. That is also true about all members of the same species. Of human now, right?
Yeah. And as far as I understand, even anthropology and some sort of data of that kind would sort of point to Neanderthals being very human-like, so to speak, not only genetics data. So yeah, that could very well be true, I guess. Not being an expert at all on this, but that I as you say, interbreeding, that's a strong point in favor of a common ancestry between Neanderthals and humans.
But do we actually have like a full set of chromosomes like intact of Homo erectus? That seems really weird if we did.
No, we don't.
So how do we even know how many chromosomes Homo erectus had?
They don't have whole chromosomes. They have fragments of DNA they can sequence and then piece together.
Do you feel that that's trustworthy to arrive at a number of chromosomes?
I don't know whether they know how many chromosomes Neanderthals have. I don't know. Don't know the details.
That was my point in relation to chromosome 2. If it were demonstrated that, say, Homo erectus also had a fused origin of chromosome 2, then Ola would have to propose, I think, on his model that Homo erectus is one of us. It is Homo sapien.
Oh yeah, but they don't have any DNA from Homo erectus, only Neanderthals.
Mhm.
So
yeah. Elizabeth Schofield in London says, how do we know that Neanderthals interbred with humans? Is that a recent discovery, and what data was it based on?
Yeah, I mean, I think the last 10 years, ancient DNA has been typed for various archaic species. I mean, descendants of Homo erectus, according to the common descent model, and the Denisovans and the Neanderthals, and those studies have sort of generated that conclusions. It's just the last 5-10 years, a number of studies that has sort of pinpointed that probably there was interbreeding between humans and Neanderthals. And according to the common descent time scale, that happened like 50,000 years ago or even more recently. It's like when you compare the DNA profiles of humans alive today and Neanderthals, and then you sort of fit a mathematical model with sort of inbreeding into it. It's such a model fits well. Then I think I haven't read the details of how they came to that conclusion, but yeah.
Peter had his hand raised. Did you want to say something, Peter?
Yes. First of all, hello. Great to be joining you.
Hello.
Peter.
Something about hello. Just wonder if you can be saying something about created diversity, because it seems to me that a key part of this is how that might turn out in the data.
Yeah.
It's a bigger part than perhaps we've given time to at the moment, as I understand it. And how might we discover whether that is a valid assumption? How can we determine that empirically? Obviously, haplotype blocks are a very key part of all that. But can you just expand that a bit further, please?
Oh yes, that's a very good question, Peter. I think creative diversity is like another parameter that we could vary, because the question is how much, if there was creative diversity, how much creative diversity is there? And that is coupled to how old is humanity. So in order to explain the DNA variation we have today, we have the number of mechanisms that removes variation, but we have two that generates variation: mutations and creative diversity. And for the mutation, then we sort of more or less, today it's possible to estimate mutation rates. So then that rate is more or less fixed because we can estimate it. But creative diversity, we cannot sort of estimate that by itself. So that is more or less a free parameter we can vary in order to fit data. So that means if humanity started recently, then most of the variation is due to creative diversity. And then we sort of vary the amount of creative diversity so that it fits data the best. And then the amount of creative diversity will then more or less match the amount of diversity we see today. Whereas if humanity is old, then the amount of the germline mutations come into play a lot more, and then we can still vary the amount of creative diversity. So I think that the amount of creative diversity will be come out from the model that fits data the best. But that is also coupled to the question of how old humanity is. So if humanity is very young, then there is a lot more creative diversity, because then we basically see from these blocks, haplotype blocks structure, then we basically, these 4 building blocks, well, they are basically the 4 chromosomes of Adam and Eve, and then we can more or less compare them and see how much creative diversity there is. And then on top of that, there is a smaller germline mutation variation, so to speak. So that, but if humanity is old, then those 4 building blocks have been scrambled much more by all germline mutation, or it's much more noisy, so to speak. So it's a bit harder. Yeah.
So how much freedom do you think there is to identify the empirical evidence for created diversity?
Yeah, I am sort of hopeful, because if one because of these haplotype block structures, and many haplotypes, many blocks come in 3, 4, or 5 versions, then we can think of all versions of those blocks we see in humans today. They belong, if we look at a certain block, say that there are 4 copies of them. So each one of us, we have 2 copies of a certain chromosome, a non-sex chromosome. So we have 2 copies of that block. Those 2 copies belongs to one of these 4 classes. And so it's like all humans in the world, their copies of that block belong to one of these 4 classes that come from Adam and Eve. And then we sort of can, it's like putting all these blocks from all the humans in the world into these 4 classes, and then the differences between the center points of those classes, that gives the created diversity, and the within variation within each class, that gives the amount of germline mutation. So for that reason, I'm quite positive about distinguishing created diversity from germline mutations, thinking in this block structure way.
I mean, that seems to me to be a very important point, because some people who are cynical could say, well, or skeptical, however you want to put it, could say, well, of course, if you have this imaginary idea of created diversity, well, you can produce anything. So the question would be, how can you separate out effectively common descent from a unique origin pair by finding something in the empirical data that will actually give you a better answer with one method as opposed to the other?
Yeah, that's an excellent point. Yes, because then that would go beyond. I mean, the first step would be we can sort of fit a unique origin model, because we have many parameters to sort of vary. But if we can also sort of look at the data and say that it sort of makes a unique origin model more probable in that way, that would be great if that was possible. Yes.
Because earlier on, I think Cherise said that maybe do we have enough data to give us and too many equations? Do we have enough data to actually give us unique solutions to these multiple equations?
Yeah. That it could be. I mean, potentially there are so many parameters to vary in these models, so we can have 2. So probably one has to start with a simpler model for unique origin, and vary to have a sort of get a crude idea of the history of humanity, and then given that, one could add more structure to it to fine calibrate, so to speak, because otherwise there is a big risk of having too many parameters, because in population genetics it's often very difficult to distinguish the different histories from each other. For one example is that if we know that a certain colony was created, people moved to a new region, but did that happen all at once, and then the 2 regions were isolated, or did it happen gradually with migration taking place over a longer period of time to this new region? Well, this is one example. Those 2 scenarios are very difficult to distinguish from genetic data today. So that's one example. So it is, so that's sort of a difficulty built into population genetics, because we only have data of today, or we used to have that. Now we have some ancient DNA data as well. But so that is a inherent difficulty of population genetics.
Can I ask you a question about that?
Just before Anne's been waiting because her husband wants to ask a question. So.
Oh, sure.
To him.
Hello. Hi.
It's a little off topic.
Yeah.
Go ahead.
Oh, the question is, Hössjer, are you using a pseudorandom number generator?
Yes. Yeah.
What kind?
Well, actually, we haven't sort of. I think Colin Reeves has written an implementation, so I don't know what kind of random number generator he has, but certainly yes, because you are quite right that our method is based on first choosing history and then simulating genetics data many times from that history and fitting it with observed data. And then in those simulation rounds, we need a pseudo-random generator. And that's not at all my expertise, but I guess that could be important how to choose that.
I'll have to ask Colin.
Yeah, okay.
Yeah.
So just to make it clear, I want to make sure that I've got it clear in my head, Ola, that it's possible for us to look at worldwide or geographically restricted haplotype blocks and sum all the variation within those blocks and then look for variation that's shared in common. Everybody has that variation in 4 different sets.
Yeah.
And then that's what's the original created diversity, and the stuff that's not shared in the 4 sets is new. Is that what you were suggesting?
Yeah, exactly. And that would be an extremely interesting project itself, because because that could be a paper of its own that doesn't need to include modeling of history, but just saying that now let's just look at one specific block and then cluster all humans copies of that block into 4 groups. If there are 4, I guess some blocks might have 3 groups or 5 groups. And that's because there is some statistical uncertainty on how to estimate those block boundaries, and that might also be the reason why the number is 3 sometimes and 5 sometimes. But exactly as you say, I think that would be a very, and Peter was into that as well, that would be a very important problem to write such a paper, and because there are thousands of blocks. For each block, we could sort of cluster into these 4 groups and have the between group variation that is created diversity, and the within group variation that is germline mutations. And then we repeat that for all we have thousands of blocks, and then we could sort of average and see the average amount of created diversity and the average amount of germline mutation. So that could as well be a sort of preliminary step in order to guide us what parameters to choose later on.
I agree.
Therese, do you want to go ahead?
Yeah, I was just going to ask about whether the existence of what's called a spline. I learned this when I was reading the African Genome Project's paper, and they looked at they did principal component analysis looking at the SNP profiles with respect to geographical locations, and they were able to, among other things, corroborate the Bantu migration and the Bantu languages, right? And they showed this thing which literally looks like, each data point is a person. And they show, like, in this 2-dimensional or maybe even 3-dimensional plot, like a linear progression of spots of the same color, and the color is from the same, like, tribe, I suppose. And that showed sort of like this geographical spread of as the DNA profiles are changing with respect to geography. And that you can do with animals that, let's say, have migrated to like a higher altitude, and now there's a bunch of alleles that have been selected for their adaptation to higher altitude. You can see this if plot that the allele profiles with respect to altitude, you know? And so honestly, I mean, I can't explain exactly why I'm kind of skeptical of the migration model at all. That's a whole other story as to where that skepticism comes from. But shouldn't there be like evidence in that respect of like this whole like when I look at the 1,000 Genomes Project or whatever, they have like categories of, okay, here's European, here's Asian, here's South American, here's the African, and it's like, well, shouldn't there be like this gradual progression of SNP profile change going from, or you know what I mean? Otherwise, what the model is just like total bottleneck, and then like genetic explosion of new SNPs, and then another bottleneck and another explosion. And I just don't really find that convincing as a historical model for how the separation, you know, I want to see like the actual gradual progression. Now is my point.
Yeah, and that's an excellent point, because as you say, the progression geographically with dispersion and migration is probably gradual. So, but I guess it's about complexity of model, number of parameters. As a first start, one could think just as if you have a continuous function that varies gradually, you can approximate that by a stepwise function. So maybe as a first crude approximation, one could say that rapidly people move from one continent to another just to get fewer parameters. But then there was also another point, very important point in what you said, that those principal component plots where you sort of cluster different geographical regions, that's actually a fourth summary statistics. Because my three summary statistics were looking at the total variation, that is nucleotide diversity, looking at the allele frequency spectrum, that is how the frequency varies when you look at one SNP at a time, and then the block length has to do with looking at several regions at the same time. But none of these include geographic variation. So a fourth summary statistics that sort of can be built into this, and we do mention about this in the papers, are based on geographic differences. And you want to capture that in the model, so you would need those kinds of summary statistics as well, that sort of compare DNA of Africans and Europeans and so on. And that can be done in many ways, but principal component analysis, as you said, that is one possibility. So we need to fit that as well into the model and have those kinds of summary statistics that takes geography into account.
But Ola, in order to validate the model, where do we need to go first? I mean, there's a plethora of opportunities out there, and there's so many things that one can adjust. But in order to establish some sort of empirical validity for the model, where do we need to start? Because I mean, otherwise this becomes a huge project.
Yeah.
How does one go about establishing that there's a difference between the ideas and the reality?
That's an excellent question. I think one possibility is to start with a smaller genomic region, and one possibility is to start with one geographic region, only Europeans, and one possibility and one possibility is to start with quite crude historical models first, not to have too many parameters. So I think one has to start with simple models. I mean, Anne, she was interested in the HLA region. I mean,
that's for later.
I guess, because it's more complicated, because we have selection and gene conversion and all kinds. Yes, you're right. So maybe one should start with quite a simple region that when not so much is happening.
Yeah, one chromosome is my suggestion, and also one chromosome.
Yeah.
Also, we have to get the programming done.
Yes, that's true. That's true.
That's where we have to start.
Oh yes, and also I think what came up now during this talk to have this sort of look into these haplotype blocks and
Yeah.
cluster them
I think that's a good idea.
within blocks, within block copies, block versions, and between them to separate germline and created diversity.
Yeah.
Because people that sort of believe in common descent, they have explained. I mean, it's quite remarkable that the blocks are so long, because that sort of indicates well, that sort of indicates that there have been that humanity is quite young, and there have been quite few ancestral recombination events. But the explanation today, in order to explain the haplotype block structure, is that we have recombination hotspots, and that might still be true, that there are recombination hotspots, but then the reason is that there are many historical recombinations, and humanity is old, but they happen at the same place because there are hotspots for them. And it could be a combination of both, that there are hotspots, but there are that's another sort of issue in this.
Well, wouldn't you predict, Ola, that if there were hotspots responsible for the long haplotype blocks, that would get rid of or mask the signal of created diversity? That it would look like there was no created diversity because everything would be shuffled. You follow?
Yeah, if there was, yeah, you're right. Because if there was, you mean even with a common descent, it could
Well,
look
even with a unique origin, if there were hotspots and in long age, it would shuffle things so much that you wouldn't be able to detect a created diversity signal as separate from the new mutations.
Yeah, that's true. That's true. And that's why that's an excellent point, because in that sense it would be possible to sort of have a first preliminary test for the age of humanity by doing this comparing diversity between the block copies and diversity within each block. Because if there is more variation between the blocks than within them, then we don't have this blurring, and then that points to a young age of humanity. Whereas if there is much blurring within the blocks, then that is, as you say, then we that pinpoint that sort of indicates an older age of humanity. Yeah.
But until you can actually run a model and get some numbers out to look at how significant the differences are between the common descent model and the created diversity model, we have no idea really the scale of what we're looking at, do we?
No. Yes, because there are many things we have to learn now when we start fitting the created diversity model. Yes, but we sort of want to fit those summary statistics, and maybe we should start with the simplest ones, nucleotide diversity and allele frequency spectra, and just to learn things. But on the other hand, these block lengths are very interesting in their own right. So I mean, yes, because the unique origin models, they haven't been sort of tried before, so it's like a new world opening up. So
absolutely,
yeah.
I mean that is the thing about this model, which is almost so way out, isn't it?
Yeah, exactly. And I should correct myself, because John Sanford and Robert Carter and Nathaniel Jansen, they have written some very interesting papers on this. So we are sort of continuing their work, but it's still just that people have started sort of work on this subject. Yeah.
But what do we think is the first thing to be done? I mean, and how do we put that together? Because we're sort of on the threshold of a new way of looking at something, and how do we move this from a theoretical concept to something that's got some real world data that enables us to assess the reality of the value of the direction we're headed in?
I think we need a bioinformatician.
Yeah.
Someone who's good at analyzing sequence data and doing the haplotype block analysis.
Yeah.
And also someone to help Colin with the programming.
Is Colin on this call?
No.
I sent Colin a note about this to say this was on. I also sent a note. Is Chris Shaw on here?
I don't think he came in now.
I sent Chris a note as well.
Yeah.
Okay.
Yeah, we'll need to have Chris Shaw on here actually at some point.
And one comment about the haplotype blocks. It was, in fact, looking at HLA diversity haplotype blocks that got me thinking about created diversity. Because what the data showed was that there was an incredible distance between the four lineages, the four haplotypes, you could call them, the four lineages of the HLA.
Yeah.
And very little diversity within each lineage, indicating a young age. Very old age, apparent old age from the separate lineages, and very young age for the more recent stuff. And the disparity was so clear. And I looked at the lineages, and they were supposedly going back 30, 40 million years with no recombination, no exchange of information. And then a sudden explosion of apparent new information. And that's when it first occurred to me that there might be something special going on.
That's a really crucial point. Yes. That's.
I think we had a conversation about that, and probably in 2013 or something like that. I remember.
Yeah.
Oh yes, in Copenhagen. Yes.
Yeah.
It was. It's still something I find intriguing that a region of the chromosome that's supposed to be extremely polymorphic. Very, very polymorphic. Literally hundreds of alleles, thousands of alleles, and it all traces back to four lineages.
That's good data to encourage us to keep moving, isn't it?
Yeah.
Oh yes.
I mean, it's qualitative rather than quantitative in the way that you would expect as a mathematician, but it's certainly confirming data or encouraging data, not disqualifying data, I think.
Oh yes, certainly.
Right.
And I guess one could sort of work on two parallel tracks. Someone programming up, continuing programming up the algorithm and fitting different historic human history scenarios, starting with simpler models and going to more complicated ones. And the other parallel track would be to look at these haplotype blocks and doing some descriptive analysis that sort of gives some preliminary ideas, because that would help the other work, because that would give us some idea how much creative diversity do we see, and those results might come out more quickly as well.
Right.
I think Steve Meyer's coming to England at the end of the week, but unfortunately I should be going off to Poland over, so I shan't get a chance to see him.
Which part of England is he in?
He's coming to London on 18th.
Oh.
He's coming to Cambridge, I think, Jonathan, on Saturday or Sunday.
Okay.
Send my regards.
Yes, indeed. I shan't see him, I'm afraid.
How long is he in the country for?
Sorry?
How long is he in the country for?
I don't know. You could email him and ask him.
Cool.
The 18th. What day of the week is that? Okay. Shucks, I was hoping to meet with him.
What?
I was hoping to meet with him on Thursday.
Oh, well, I think he's flying out of Seattle on Wednesday evening.
Okay.
They're both, he and his wife, Elaine, is it?
Elaine, yeah.
They're both coming over, I think. He called me on Saturday.
Yeah.
Anyway.
Yes, sir. It's Friday.
Very excited to be having this conversation, I have to say. And nobody's jet lagged, which is rather nice too.
Yeah. So I think those conversations, they are, that's a very good created atmosphere to have this Q&A afterwards and sort of try different ideas back and forth. So,
yep, we do these every week. So there's a
very
whole catalog on the Apologetics Academy YouTube channel of about 60 plus videos now. So we have quite a lot of recordings.
So how many people attended today?
Total, we got 18. And that's on Zoom. And then there was about 15 to 20 following on Facebook as well.
Oh, that's good.
No,
yeah.
Great.
And how many are left?
Currently left on Zoom, we have 12. And on Facebook Live, we have 4.
Okay.
I have more questions if you guys are up for it.
Yeah.
It is getting late for Ola, so only if he's...
No. This has been great with this Q&A time, and so go ahead. Yes.
Well,
yeah.
Kind of what you touched on it already, but I wanted to get further clarification about the SNPs. Well, the created diversity. And okay, so what type of diversity are we talking about here, right? Are we talking about neutral, so that whatever the behavior of that locus in the genome is, or not the behavior of it, but whatever the system of the cell recognizes about that section of DNA is essentially, the same? Or is it like, does it provide some new biological meaning at that, because of that change? And then how does that relate to what you had said about the avoidance of the inbreeding problem?
Yeah. I think because the selection is on the phenotypes and not on the genetic variation. That means that most SNP variation by itself has sort of quite a small selection effect, but those that have are, I would say, then slightly deleterious, but most of them are neutral then. So I would say that if we collect all the several million SNPs there are and do various statistics of them, most of them will, I guess, be neutral or slightly deleterious. There might be certain regions where there is a lot of selection, but those regions will sort of constitute such a small region of the whole genome, so that as a first approximation, one could even think of most of the SNPs being neutral. But of course, that depends on whether it affects the codon affects transcription into amino acid and changes the protein and so on. But but since we look at genome-wide statistics, the idea is that as a first approximation, it could be quite good to view all the SNPs as fairly neutral.
I thought of the created diversity as being neutral or beneficial, and therefore having no impact on inbreeding. In fact, that's the requirement, is that they all be, each set of chromosomes has its own set of variations, and as they as the population grows and there's inbreeding, it has no negative effect because those variants are either harmless or beneficial. It's only after you start to acquire mutations over time that those new mutations have the potential to be deleterious.
Yeah, I totally agree. That's an excellent point. So the hypothesis is that the created diversity does not contain harmful variants, but then the other ones that come along, that they are either neutral or slightly deleterious, and the deleterious ones come as germline mutations. Yes, that's a very good point. Yes.
Okay, so I understand that part about how inbreeding, if you have inbreeding and then you have like a homozygous deleterious mutation from inbreeding, like that's bad. That's why inbreeding is bad. But I don't see how the created diversity impacts that issue at all, unless the idea is that it mitigates it somehow by like some sort of epistatic control. Like, so if you have, I can't even think of like how the interaction between the pre-existing created diversity and new diversity that because of inbreeding, new deleterious diversity that gets, collected in the same genome because of inbreeding and thereby causing all we know that is bad about inbreeding, like how that's a mitigating effect. And that leads to my question of what would be the purpose of created diversity, like in and of itself.
Yeah. The purpose of created diversity is to generate diversity between humans in a positive sense. I mean, just so that we are genetically different.
But that just is like a tautology kind of like it's like you create it so it's there, but like why do you want it to be there? Like why is it important? You know what I'm saying? Like in terms of survival or fitness or whatever. Like.
Yeah, I think yes. I mean, one can first think of it as a sort of God creating diversity because that is sort of beautiful in itself. But then you could also think of it in terms of microevolution. It's one purpose of having diversity in a good sense is it's easier for a species to adjust to environmental changes if there is some variation. So that could also be a way. I mean, for instance, if temperature changes and so on, it's good to have some diversity. And but the origin, as Anne said, the original diversity, according to the model, does not contain harmful variants. So there is no problem then with the inbreeding depression in the beginning. It's only when those harmful germline mutations accumulate and, as you said, appear in homozygotes, so that you get the harmful copy from the mother and the father, then you get the problem later on. So I think that the purpose of creating diversity with no harmful variants is to have diversity in order to have it for its own sake, and in order for it to, that also gets sort of makes it possible to adjust to environmental changes. And so those two purposes maybe. Yeah.
And do you agree with that?
I think for adaptive purposes, it's going to be good to have variants available in the genome, available for as humans migrate out from either Africa or Palestine, whichever it is, into new habitats. There's going to be a need for different phenotypes to appear. We don't all look the same. We don't all have the same abilities, and I don't think of that as being necessarily the result of harmful mutation. That some of it could have been designed in from the beginning as potential that could be realized over time.
Yeah.
Okay.
And it could also be that if we start with creative diversity, and then quite soon, after a number of generations, humans started to move to different regions, and those different subpopulations happen to have different genetic makeups, and that from the start sort of, and it is that part I mentioned, that colonization and isolation, they sort of generate genetic differences between regions. So the genetic differences we see today, so then could sort of be the fact that the creative diversity plus isolation, colonization and isolation created regional differences. So that's another sort of effect of creative diversity, to have regional differences.
There was a couple more submitted questions. Jacob Guthrie says, this may be a bit off topic, however, has the percentage of DNA similarity between apes and humans been settled to a particular percentage, i.e. 99%?
At the moment, it's about 5%. Estimates are the SNP variation is a little bit more than 1%, 1.08, I think. And there's between 2 and 3% difference due to insertions and deletions, and a little bit more here and there. The averages that I'm seeing in the literature are about 5%.
Mm-hmm. And Yaya Snow, our Muslim apologist friend, says, has Dr. Ola got a recommended book or resource to learn more about linking the Adam and Eve story to a more scientific view?
You mean sort of, I don't know, you mean more specifically about this question of population genetics, or broad, or apologetics in a broader sense? I think I mean John Sanford's book, Genetic Entropy, that sort of he's a population geneticist, and he gives some of the arguments about this accumulation of inbreeding depression, and he phrases that as genetic entropy increases over time, and so that's one good book to start with. Although and he and Robin Carter, they have sort of written about this fitting human histories as well, but that book doesn't contain that. So I don't know if there is a book out yet that sort of has this discussion about different human histories. But there are some books like John Sanford's that deal with population genetics and impacts for talking about weaknesses of common descent approaches using population genetics. So I would say John Sanford's book Genetic Entropy is one.
Yeah.
I
would add to that another excellent book on the subject is Science and Human Origins, that Anne Cager is a contributor to, along with Casey Luskin and Doug Axe. It covers a lot of that material, and also covers a lot of stuff on the paleontological record and other things.
Mm.
Yeah, Science and Human Origins. It's a really thin book and not too difficult to read.
Mm-hmm.
Yeah, I agree.
There's a new book out called Christianity and Science, but it's a huge compilation of essays on different topics having to do with relationship between Christianity and science, and a lot of it has to do with evolution, topics related to evolution. It has articles written from all three sides: theistic evolution, ID, and young Earth creationism. And they have some articles are standalone, some of them are two competing views. So it's a good resource if you have enough money to spring for a copy.
Yeah, there's the bit here, dictionary of Christianity and science. It's really really good resource in different topics.
Can I
Jonathan, I'm sorry. Excuse me, Sherry. I'm going to have to. No, I'm going to have to. Fast thing though is I'm going to have to back out. I'm afraid because I've got more.
Thank you very much for your questions and contribution. Thanks a lot.
Lovely to see you all. Thank you.
Yeah.
God bless.
God bless.
God bless. Bye-bye.
See ya.
Bye, Peter.
Bye-bye.
Bye. Bye-bye. Bye.
Okay, I think I have a prediction for you guys to test. Okay, if it's true that the created diversity was not just purely neutral phenotypically, or it had some sort of phenotypic potential in there somehow, then that means that the children of Adam and Eve would look so different from them because you're combining things that they don't even have. Like, so Adam's giving something and Eve is giving something, and you're making something that is totally different from both of them. Because if in the combination of these diverse chromosomes and the way they interact, you would expect there to be a new set of phenotypes. And you would, unless they're perfectly neutral, then, the whole theory about what the whole reason they're there for goes away. But if they're there for that reason, and this is like sort of the driving force for phenotypic diversity across the human population that we see now, or not the driving force, let's say more like the primordial soup for it, like where it emerged from mostly, then that, go ahead.
That scenario would work only if each what do I want to call it, each SNP was dominant. If it were the case that you wouldn't get a phenotype until you had 2 copies of the same SNP, then it would take a few generations of mixing in order for you to start to have chromosomes carrying the same alleles at the same position.
Okay, that's a good point. All right, well, let's just say they're dominant then. So let's say they're dominant. And what you would get then over time, what the prediction would be is that the more generations you get to, the less different the children look from their parents. The less likely you are to get different looking children from any like random couple you pick in the population.
Once again, I think about it differently. I think of it as them being like recessive alleles, and also pleiotropic. You have to have more than one in combination to get the phenotype. So it's really working out over time of the potential for differentness that will appear as groups migrate apart, and you start to get distinctive combinations appearing that are associated with different groups in different locations.
Yeah.
So, but as far as if inbreeding is happening by necessity at the very beginning, wouldn't you get these recessive alleles coming together and homozygous?
The thing about the first generations, as I understand it, and Ola would know this better than me, the theory is that it's an explosive process.
Yeah.
We have rapid multiplication of the population because everybody's long lived and they produce lots of children.
Yes.
And generations overlap. So population grows rapidly.
But that's still inbreeding though.
Yeah, but the diversity is maintained. It's not like a situation where you have a bottleneck and diversity is lost because of inbreeding.
Yeah, that's an excellent point. That in order to really lose diversity, you have to have quite a. If it's just a very short bottleneck, and as Anne said, the population size increases rapidly, then you don't lose many variants. Actually, and even if you have then the flood and Noah, that's also a very short bottleneck, because then you are down to like 8 persons, but that's also then very rapid population growth after that.
But I'm not suggesting for bottlenecks, though. I'm suggesting that within the population, you'll have a number of homozygotes, right? They don't. So what the impact of that is who knows? That's like kind of the you know, what I'm suggesting is where you'll end up seeing something different from when you have heterozygous people. But so as far as the bottleneck, that's saying that the effect of being homozygous is, you know, you have either a selective sweep or something, or, you know, because the population is small, these guys are drifted out, or I don't know that what I'm saying, maybe I'm misunderstanding some fundamental thing here, but like if you just keep reproducing and you keep like inbreeding, like there's a high probability that if you started with 4 chromosomes that you're going to pair up like some of those down the line, and that's going to give you, if this mechanism that Anne you're talking about is the case, like you're going to see that diversity really quick.
It's possible you see it really quick. It's also possible that you need to have the right combinations of homozygous genes together, that it's not just one gene determining a new phenotype.
Yeah.
So you might need to have gene A and gene D and gene J in order to see blonde hair, for example, or something. You know, it's not a one-to-one correspondence between genotype and phenotype.
Sure.
Yeah, and I should correct myself when I said that the model sort of should incorporate that the created diversity is neutral. What I mean that it could still affect phenotype, but I mean it should not affect the phenotypes in a bad sense. I mean, it could generate different kinds of eye color and so on. So in that sense, generate diversity. But if none of this created diversity generates sort of increases risk of having some inheritable disease and things like that, then it need not be a problem. You're right that even though the population increases quickly, there might be some individuals that are homozygous. I mean, grandchildren of Adam and Eve, for instance, their 2 parents are then siblings, but today that would be not be a good thing. But in these first generations, even though the grandsons and granddaughters of Adam and Eve, some end up being homozygous at certain SNPs. If it's still the case that this variation doesn't produce harmful phenotype variation, it might produce phenotype variation, but then it need not be dangerous to have a recessive to have the homozygous, because even though you have 2 copies of just one allele, that need not be harmful, because none of the alleles are coupled to harmful phenotype, so to speak. I mean, according to such a model. Yeah. Do you agree with that, Anne? I mean, that's sort of
yeah. I think the question you were asking, Charisse, is why would there be created diversity in the first place, except as a fudge factor for the population genetics model?
Yeah. Like, what's its purpose? Like.
Yeah. And I think it really is there for diversity's purpose, to emerge down the line, whether it emerges rapidly or more slowly over time.
Okay, then here's another prediction. Like, if that is a purpose that creates robustness in the species, would you predict that all species have this?
Yes.
Yeah.
Ooh.
Yeah.
Yes.
And
I like that. I agree. I think so too.
Yeah, I think from a Christian point of view, it would be a beautiful way for God to create diversity. I mean, it's beautiful to have diversity within a species. It sort of gives variation, and this is then a very neat way of doing that. Yeah.
Yeah, I think it's very functional though too, because here's this really annoying thing about evolution, this idea of fitness, or yeah, you know, you're fit because the survival of the fittest, right? You're adapted to your environment, and that's why you survive. But at the same time, like, any change that you make to that genome, like, it sort of changes your relationship to the environment in a way that you either survive it or not. And so there really isn't sort of like a metric on there's not really a good metric, I don't think, you can use to explain that phenomenon of fitness, even though we use differential reproductive success as a number you can count. But it's, to me, is like really doesn't make a lot of sense. But let's just say, imagine like a completely static and optimized and ideal environment. And there's no such thing as migration out of it. Then there really is no need for diversity. And right, because like what you would want is to create this optimized genome for this optimized environment that's static, and have the genome be static as well. But like, if part of the lifestyle of the thing is that it's going to, you know, spread through several kinds of environments, if anytime you like got out of like this band of environmental factors, like, you know, the next generation went extinct because it just absolutely couldn't handle it, then that would probably be really limiting, I guess. So maybe this whole instilling created diversity thing could be like front loading the possibility for migration.
Yeah.
In a fluctuating environment.
I see parallels with what you're saying in microbiology, in just growing E. coli in the lab. If you grow a culture, say E. coli, on citrate medium, like Lenski's done for generation after generation, it starts ditching genes it doesn't need so as to be really efficient at growing in that environment. And once gone, those genes are gone, it gets really efficient at growing in that environment, but put it out in the wild, it won't work. Same with yeast. Budding yeast has a sexual reproductive cycle and a budding cycle. Excuse me. And you can grow yeast under conditions where it does not undergo reproduction, sexual reproduction for generations, and just keep doing that. It will ditch the ability to reproduce sexually. Just for efficiency's sake, because it doesn't require it anymore. But if you look at organisms as they are, they have all kinds of potential that they don't use all the time, and yet they're maintaining those pathways that they don't necessarily need, but they're there, And it's only when you put them in a very limiting environment and it becomes beneficial, so to speak, to get rid of the thing, that they lose it. So I see there as being a lot of diversity inherent in each organism. We talked about phenotypic plasticity when we were in London.
Oh, yeah.
Yeah.
So it's there. It's
So
built in.
what does that look like in the human population then? I mean, do we ditch genes over time, or, you know what I mean? Is there a corollary there with the human population?
We're not living in a petri dish, a monoculture.
Populations are.
We're not living in a monoculture, really.
Maybe the equivalent is those moms who bleach everything in the house, and then their kids end up having, like, you know, autoimmune disorders because the immune system is like, I don't need, you know, I never got any practice, so now I'm just going to attack you.
Yeah. You've taken away my purpose in life. So.
Right. No, that's really, really interesting.
Yeah. It's conservation biologists, they have sort of criteria for they want to, in the short run, prevent inbreeding from accumulating, but then they also have long-term goals to have the species adjustable for environmental changes that might happen. So that also reflects this discussion a bit, that there are sort of two goals that are important, or two purposes of diversity in that sense. But the inbreeding impression, that only sort of that's only comes along if some of the variants are harmful.
Right. The original HLA story got me in. One of the other things that got me into thinking about is it appears that there's a mechanism in place to generate diversity really fast in the HLA region. And we're talking, you know, hundreds, thousands of alleles in the course of a couple of, say, 200,000 years. Huge numbers of alleles in different genes involved in the immune system. And it struck me that this makes sense from a design point of view, that you want to be able to protect organisms that go through bottlenecks so that they can regenerate their immune diversity really rapidly.
Yes.
There should be a mechanism provided for. And it was one of the things that I wanted to research. Never found a immunologist to help me with. But my prediction is that the mechanism is there, and it's going to be similar to the immunoglobulin splicing, that the same enzyme is used, only it's used in the germline.
Interesting.
That's a really fascinating suggestion.
Yeah.
So you're saying that what that would be there to mitigate is if you had a human genetic bottleneck through migration of some sort, that would actually be, oh, I see. So this would work to restore balancing selection kind of thing, like where you have to have a standing amount of variation in the population. And so there's some mechanism in the germline to recreate that, or to make sure that that's kind of always there in any given population.
Right. There's evidence to suggest that this is actually going on in the literature. Human populations that get isolated, such as South American tribes, they generate new versions of new alleles of these HLA genes within, I don't know, 1,000 years. It's really remarkably fast.
Mm.
And there's one paper I found where they did a comparison of the alleles present in a father versus his sperm. And they found there was a mutation rate on the order of 10 to the minus 3 to 10 to the minus 4 in these genes.
Oh, that's very high. And then that's then a sort of beneficial mutation rate.
It's a designed mutation.
Yeah.
Mm.
And it's important in protection against disease. It's also important, we need diversity in the HLA for fertility, because moms carrying babies with the same HLA type don't do so well.
Mm. Yeah, I heard. Did you hear about that study a while back where they gave women worn T-shirts of a bunch of men, and the ones they were attracted to, the smell of, were further away from them in terms of their, I think it's this HLA diversity.
Yep.
And so it was like there's this sort of primal drive to mate with people who are, sort of different
Mm-hmm.
from you.
Yep. You need the balancing selection. The more diversity you have, the better you'll do against disease challenge.
Mm-hmm.
Interesting.
Quite a chicken and egg problem then, if you think about it outside of the design paradigm. Like any kind of selection that happens on the population scale that, that's beneficial for the population is really hard to explain, I think, in neo-Darwinistic terms. But I think that this really, I don't know, I'd be really interested to look at that, the enzymes involved in generating that.
What the molecules are?
Yeah, the specified mutations rates in those genes themselves, that's really fascinating.
It's a protein that's got about 6 exons, and exon 2 is where all the variation sits. And it varies, it's over just about 200 bases. And
the HLA gene or the enzyme that creates the diversity?
Oh, the enzyme that creates the diversity. I forget what it's called. ACAR, something like that. I don't recall. I don't remember the details.
I can look it up. But that there must be some sort of signal to know, like, okay, when you're replicating this part.
There is a signal known in immunoglobulins. Nobody's found a signal in the HLA genes.
This looks like something maybe Josiah can look into.
Actually, I was going to ask Chase Nelson. He was going to help me with the project for a while, but he got too busy. I need an immunologist and a computer whiz to do the analysis. But I've already got enough to do, so.
All right, shall we wrap up there?
Yeah, this has been really good, you guys. I'm really excited for the next steps.
Yeah, thanks for the questions and discussion. It's been, I've learned a lot, and
thanks for joining us, Ola.
Thank you all.
And you.
Have a good rest of your week.
Okay.
Yeah.
All right, bye, you guys.
Bye-bye.
Bye.
Bye-bye. Bye.